Species as stressors. Heterospecific Interactions in the Cellular Stress Response under Global Change is a research paper focused on the effects of cellular stress responses and what causes them. The purpose of the experiment in this research paper was to determine if there are more cellular stress responses between two different species as opposed to two of the same species, and how much that difference is. When we think about the effects of stress in our body, we often associate it with negative physical effects, like an increased heartbeat or shortness of breath. However, while these factors are crucial to monitor, stress also affects organisms on a molecular level. When faced with situations that induce stress, organisms undergo a cellular stress response, CSR, or molecular change to counteract the effects of the stress. An example of cellular stress response is a heat shock protein, which protects cells from damage during protein folding as they undergo stress. Will interactions between P. manimaculus and P. sync-types result in CSR due to interspecific interactions? Crabs were collected from South Florida, and a 2-week time period was used to allow the crabs to adapt to their new environment. Thereafter, 3 different types of trials were set up, in which one crab was placed with 4 other crabs of the same species, a different species, or alone in the case of the control. The crabs of interest were painted and observed for an hour for agonistic interactions, and this was repeated 5 times for each species and each scenario. Water temperature was kept at 13 degrees Celsius to prevent cofactors.
After this, focal crabs were frozen with liquid nitrogen and gene expression was run for 6 genes. P. sync-types occupies a higher, warmer area of the intertidal zone than P. manimacules, and if overall temperature was to rise even more, they would have to resort to occupying a part of the same habitat. This will result in more overall interactions between the two species, and this experiment showed that there was no effect from species on gene expression. Since this will result in the species interacting, it is important to observe how this affects the species. The control group exhibited a small decrease in PC1 gene expression, while the conspecific and heterospecific groups showed a large increase and decrease respectively. However, the error bar was so large for all the groups that the data may have appeared to show a trend that was inconclusive. The results showed that the species did not act as a stressor for gene expression. When this data is plotted comparing gene expression of PC1 and the number of agonistic interactions, there is no correlation. While other studies have resulted in evidence indicating that species can affect each other on a cellular level, this study was inconclusive. Some reasons for this may have been that the limited observation time was around 1 hour, compared to a more realistic observation time that would occur in their natural habitat. The way species interact with one another is important in the study of ecology. Organisms can impact one another and the environment around them. They can have different types of relationships, mutualism, parasitism, and competition. Species can work to alleviate this competition for niches by using resource partitioning. In the crab experiment, it was concluded that there was no correlation between the two different species and the amount of cellular stress. However, as researchers stated, there is more research to be done in the field. There is a correlation between the number of aggressive behaviors of crabs who are of the same species as opposed to crabs of a different species.